at T cells in general, T cells, I must have mentioned previously that T cells have CD3 on their cell surface. They have CD3 on their cell surface, and they have two major groups of T cells. We have CD4s and CD8s. CD4s are also referred to as T helper cells, and CD8s are referred to as T cytotoxic cells or cytotoxic T cells or helper T cells for CD4. Okay, so again, when you look at CD4s, we have other subgroups within CD4s. We have other subgroups within CD4. So CD4, then again, we have those that are referred to as TH1, TH2, Tregs, and TH17. So we have subpopulations or subclasses of these CD4s. And we also have subclasses of CD8, and they are majorly TC1 and TC2. And so TC1 and TC2 behave like TH1 and TH2 of the CD4s. So among the CD8s and CD8 T cytotoxic cells, they are just made up of two subpopulations, that's TC1 and TC2. But TC1 and TC2 behave as TH1 and TH2. We'll see in a moment what is the difference between TH1 and TH2. Okay. So then briefly, we're talking about antigen presentation and processing. There are usually two pathways in antigen presenting. We have the cytosolic pathway or the in. So when you're talking about an antigen that is intracellular, an antigen that is intracellular, then we are calling it a cytosolic antigen or an endogenous antigen. While we have those that are exogenous, I mean they are extracellular. So we have two pathways, a pathway that is targeting antigens that are extracellular, and that one that is targeting antigens that are intracellular. So intracellular antigens, we refer to that pathway as being cytosolic, and for extracellular, we refer to that pathway as being vascular. So vascular, extracellular. Okay, so when you have a pathogen that infects cells and gets into the cells, that is an intracellular pathogen, then antigen presentation will be done via major histocompatibility 1 to CD8 cells. We know what CD8 cells do. They are cytotoxic T cells. While if it is extracellular, then the antigen presentation will be done via major histocompatibility 2 to CD4 T cells. Okay, that is what happens. So antigen presentation, we've seen that is done by either dendritic cells, macrophages, or B cells. And in antigen presentation, we require a key molecule, rather type of proteins referred to as major histocompatibility complex. There are three types of major histocompatibility complex, type 1, 2, and 3. 3 has no immunological importance. Type 1 is a groove where intracellular pathogens are presented. So remember, in this case, the dendritic cells are going to pick proteins and present them on the major histocompatibility complex. And so this major histocompatibility complex is the one that is interacting with T cells. Remember, antigens are presented T cells, and we've said we have two types of T cells. We have CD8 and CD4. So if an antigen is intracellular within the cell, then it will be presented via MHC1 onto CD8. If it's extracellular, then it's going to be presented via major MHC2 onto CD4 cells. And CD4 cells are referred to as helper T cells. We've seen that. So antigen-presenting cells internalize and process antigens, presenting them to T cells. so that then they trigger immune activation. That is generally what happens. For the interaction between antigen-presenting cells and T cells, and we've seen what antigen-presenting cells are, the three, apart from them just interacting, they require what we call, because the interaction is via what we call the T cell receptor. So antigen-presenting cells interact with T cells via the T cell receptor. And apart from interacting with the T cell receptor, they require costimulatory molecules, specifically CD28 and CD80. Okay. Apart from this, apart from their normal interaction, through the TCR, the T cell receptor, they need CD8 and they need CD80, CD28 and CD80. So in other words, what we are saying is that if you look at DCs, dendritic cells, they are going to have CD8 on them, CD28, sorry, on them. They are also going to have CD80, similar to macrophages, similar to B cells. They will have this, and these are referred to as costimulatory molecules. If they don't link, then the presentation will not be done well. So once that is done, then say, for instance, CD4 T cells are able to release cytokines to aid in immunoregulation. Okay, so we're saying CD8s are specific, CD4s are going to generally produce cytokines. CD8s, their work is to perform what we call cytotoxicity. So most likely they are going to perforate the cell, inject it with perforins, and of course kill the cell via lysis. But for CD4s, they are going to release cytokines. Okay, we saw that we have four different types of CD4s. We have four different types of CD4s. We talked about Th1, Th2, Tregs, and Th17 as being the other subpopulations of CD4s. So Th1s secrete interferon gamma to activate macrophages, natural killer cells, and CD8. So they promote cell-mediated immunity. That is what Th1s do. So it's a class of CD4s, and their main work is to secrete what? Interferon gamma. And once interferon gamma has been secreted, that is the cytokine they produce, then it activates macrophages. We know what macrophages do. It activates natural killer cells. We've seen what natural killer cells do. They also activate CD8. So the question here would be, are natural killer cells not part of the innate immune system? Yes, they are part of the innate immune system. So in the innate immune system, they perform whatever they do. And it doesn't mean that if in the innate immune system, interferon gamma cannot be released. Definitely it can be released. But this is adaptive, and so it's more specific. In this case, it means the body knows that either this is a tumor or this is a virally infected cell. So of course, natural killer cells are brought in, and that is what they do. And of course, I also CD8. CD8 behave the same way as natural killer cell. They perforate, they have granzyme, they have perforin, and they perforate cells so that those cells are then able to lyse. Th2 cells, they secrete interleukin-4 and interleukin-10, helping B cells to differentiate into plasma cells so that they're able to produce antibodies. Okay. Now remember, we are calling T cells helper T cells. That is where the name comes from. We are calling them helper T cells. That is where the name comes from. We're just saying they're secreting IL-4 and IL-10. And interleukin-4 and interleukin-10 will help B cells to differentiate into plasma cells. So, in other words, they help B cells mature into plasma cells, and once they become plasma cells, then they are able to produce antibodies. Then you have T regulatory cells, Tregs. Tregs, apart from them being CD4s, they express another surface marker called CD25, and then They usually produce a transcription factor referred to as FOXP3. Okay, so the unique thing, so CD4s are T helper cells. So T cells are CD3s. Then among T cells, we have CD3 that are CD4, then we have CD3 that are CD8. But apart from that, we are having a CD4, CD3 that is CD4, and it's CD25. Okay, so it's also expressing another cell on it. I mean, another marker on its surface called CD25. But still, that does not qualify to define it as a T regulatory cell. So to qualify it, it produces another transcription factor called FOXP3. So this is what defines regulatory cells. Now, T regulatory cells usually secrete interleukin 10 and tumor growth factor beta. Okay. Remember, Th2 are also producing what? Interleukin 10. So we can't use interleukin 10 to differentiate Th2 from Tregs. So we're using FOXP3, but other than this, they're also producing tumor growth factor beta. Now, interleukin 4 and interleukin 4 is a pro-inflammatory cytokine. It leads to inflammation. This one is anti-inflammatory. But these two, this is anti-inflammatory, this is anti-inflammatory. Okay? At least this is pro-inflammatory, this is anti-inflammatory. So these are important in B cells differentiation. But important in slowing down inflammation. That's why T regulatory cells are very important, especially in preventing autoimmunity and regulating immune responses. Because you see, you don't want an immune response to be all pro-inflammatory. You want it to slow down at a point. So homeostatically, T regulatory cells are very important. It's been discovered a few years ago. It's not, it could be around your ages, just about, late 19s, 2018 years, I think, ago. It's not a very old discovery. And this is the newest discovery, of course, Th17. It could be about 15 years old now, they're about. Th17, they produce pro-inflammatory cytokines. And how we make, because this is also, they produce interleukin 23. They produce interleukin 23. That is what makes them a bit unique. So these are pro-inflammatory. These are pro-inflammatory. These ones are just targeted for B cells, and these ones are anti-inflammatory. So those are the three subclasses. I mean, not three, four subclasses of T cells. Okay. So interferon gamma is the main pro-inflammatory cytokine. Yes, it is. Now, interferon alpha and interferon gamma do have roles. They do have immunological roles. They can be classified as pro-inflammatory cytokines, which means they trigger inflammation, although the way they are presented is not enough. So their production is not just intergamma. So interferon gamma now is more is like the main, how do I call it? It's the main cytokine that is produced to trigger inflammation, and it masks the rest of the cytokines. It masks the rest of the cytokines, including interleukins, including interferons. Okay, any other type of interferon, be it even delta, be it even epsilon, they are produced, yes. But the major one that is produced is interferon gamma. It's like if you go to any site of inflammation and you're measuring the cytokines present there, others will be there, but the main one will be interferon gamma. So they do play a role, yes, but the role is not very significant, and they are pro-inflammatory, of course. So their role is not very significant. And of course, as these molecules are being discovered every now and then, their different roles are being identified. For instance, like I've just mentioned about FOXP3, I've mentioned about T regulatory cells and how they play preventing autoimmune. This is just new knowledge that's being generated. So you find that there's one interferon gamma that is being overproduced, that it masks the rest of the molecules that are being generated, so that focus is there. But if we these molecules, we're able to see them produce. But we can't interferon gamma, I mean alpha or beta or even delta or epsilon are important molecules per se, because they are produced in very small numbers, much as they have a bit of inflammatory responses that they do. So the opposite of interferon gamma is interleukin 10. Interleukin 10. So this does the opposite of that. Interleukin 10 is the anti-inflammatory, and of course the tumor growth factor beta. These two are opposite of this. And of course, we have this being the key ones, interferon gamma being the main pro-inflammatory, but as well as interleukin 2, interleukin 12, interleukin 11. So quite a number of interleukins are pro-inflammatory. But then these are the key anti-inflammatory, interleukin 10 and tumor growth factor beta. Okay, I hope that is clear for you. So, in essence, there are very many molecules that are involved. It's just that we are focusing on the main molecules here. Right. Then let's look at T-cell interaction. So T-cell, of course, is able to interact with antigen-presenting cells. We know what is happening here in antigen presentation. T-cell is also able to interact with B cells, so that especially Th2s, they're able to interact with this so that antibodies are produced. T cells are also able to interact with cells that are supposed to be killed directly. For instance, okay, let me ask here, which type of T cell will interact with cells directly so that cells are killed? Which type of cell will interact with cells so that cells are killed? Crispin?
I think it's cytotoxic.
Okay, CD8, yeah? CD8. That's correct. Cytotoxic T cells or CD8s, they are going to definitely interact and kill. Yeah, so those are the three types of interactions we are having. Antigen presenting with T cells and antigen presenting cells, the dendritic macrophages, as well as B cells. Then B cells, T cells, and then T cells to the targeted cells. Then we have something called the B cell and T cell receptors. T and B receptors. These are just receptors where, of course, the interaction, of course, B cell will interact membrane antibodies through B cell receptor. Then T cells are going to interact with the peptides, Major Compatibility Complex, through the B cell receptor. So both of these are found in the immunoglobulin superfamily gene. They are found in the immunoglobulin superfamily gene. So of course, the immunoglobulins are what we refer to as the antibodies, and antibodies are shaped like like this. This is how they are shaped. So when you talk about the immunoglobulin gene superfamily, it's going to give you what we call variable chains, like that, light chain, variable. Okay, this is how an antibody looks like. For instance, this is IgG, this is IgA, this is IgM, this is IgE, this is IgG. We have 5 types of antibodies, and of course IgG has different subtypes, usually Ig1 to Ig4. There are different subtypes. So IgM being the largest immunoglobulin has a valency of 10. Has a valency of 10. 4, 5, 6, 7. No, valency of 20, not 10, 20. A valency of 20, sorry. So this like 5 antibodies together, 1, 2, 3, 4, 5, 6, 7. There's a valency of 20. IgA has a valency of 4. Most of these others have valency of 4 because they, IgA might be a valency of 8 because it's bound here by a J chain. So when you talk about, so what I wanted to emphasize is this thing called the immunoglobulin gene family. In other words, if you look at the B cell receptor and the T cell receptor, they look like this. They are Y-shaped. They are Y-shaped like this. That is what we mean by that. So the gene that produces the T cell receptor and B cell receptor will form a Y-like about an antibodies and their structures in details when we'll be talking about humoral immunity.